Kwanza umeanza kurekodi. Mbinu yetu ya juzi ya itatake muda mfupi sana, hizi hata 10 minutes. Usicomplicate swali kidogo swali. Ndio swali ni test tu. Ndio. Hello. Are you all in? Are you able to hear me? I can see Callistus. Are you in? Yes. Obuya? Okay, welcome. Today we are going to learn about cytology, being the first topic this term. I am Meshak Mkenda from the Department of Health Sciences. I welcome you all. The title of the topic today is microscopy. I hope you are a bit familiar with what is microscopy. Who can define the term microscopy? Yes, Callistus? Yes. Microscopy is a process of observing microorganisms through a microscope. Now, our objectives today are: 1, by the end of this lesson, you should be able to define what is microscopy. Number 2, you should be able to describe types of microscopes. And number 3, identify parts of a compound microscope. Now, just a minute. Now, are you able to see these notes from your end? Now, let's continue. I asked, what is microscopy? Someone to try? Yes. Good, Riziki. That's a good trial. Now, in our introduction, I'll define what is microscopy. It is a common practice done in the laboratories to determine various types and forms of minute organisms. Examples of these are bacteria, viruses, and fungi. Next, we are going to define the terms used in microscopy. And the first term is a microscope. Mr. Aruba, can you tell us What is a microscope? Yes, that is perfect. It is an apparatus or a device used in observation of microscopic organisms and materials in a lab. Another term is bright field microscopy. Callistus Obuya, can you try what is bright field microscopy? Yes, Obuya? Yes, it is like Obuya, I'm not getting you. Well, let's continue. Bright field microscopy is a type of microscopy where light is used to illuminate the field of view while the organism appears dark. That is, the field is illuminated while the organism appears dark. The last term is dark field microscopy. Riziki, can you try dark field microscopy? Yes, yes, that is right. Is a type of microscopy where the light rays are directed to the organism while the field of view appears dark. Now, our next slide is the parts of a microscope. A microscope has various parts, the ocular parts and the mechanical parts. The ocular parts are the delicate parts which are used to view specimens. Give us one example of the ocular parts. Anyone to raise up the hand? Yes, 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 that is right. Ocular parts, they include the lens, uh, that's the eyepiece, which has components that you use to view the organism or the specimen. We also have the mechanical parts, which are the physical parts that you can handle. And Mr. Aruba, can you tell us one of the mechanical parts you have come across through your microscopy? Yeah, that is good. Like the arm and the base, which you can hold on, and they hold the delicate parts, the delicate ocular parts. Our next slide, we are going to look at functions of parts of a microscope. One part is the eyepiece, where Riziki has just told us that it is where we place our eye to view the microbes. The other part is the base. The base is where the microscope sits on the benches in the lab. Also, we are going to see the differences between bright field microscopy, dark field microscopy, and fluorescent microscopy. Mr. Barasa, can you tell us one difference between bright field and dark field? Yes, Barasa? Yes. Yes. That is perfect. Can you tell us what is fluorescent microscopy? Yes, that is good. Fluorescent is where the specimen uh, sparks gives some sparks or uh, like a star. Let me give an example of a star. While you look at a star at night, it glitters. Specimen viewed under fluorescent glitters like a star at night. Lastly, uh, one of you to give us a summary of what we have done. Aruba, please. Now, it is like Aruba is off. Today, we have studied microscopy, which is a science of studying microbes by use of microscopes. And there are three types of compound microscopes which we have just looked at. That is bright field, dark field, and fluorescent. I'm going to leave you with an assignment which we'll collect in the next class. One, in groups of five, research on uses of parts of a microscope are shown on the micrograph that I'll give you. Thank you and welcome. Hey,